I am a community ecologist by training, and which means that I study how species interact with each other and with the environment around them to come together to form what we see as ecological communities defined by some limits of space and time. So in other words, I like to understand why species are found where they are found, what makes some of them rare and some of them common. How is it that rare species persist and are not outcompeted by the more common abundant species? So in effect, I like to, or I'm interested in the questions or the processes that allow species to coexist and maintain the diversity of ecological communities. A lot of these questions I pursue using tree communities as model systems, and which is what a lot of my work has been about largely. But these questions of species distributions and coexistence are their intellectual value and curiosity aside, are also of practical relevance in today's world. We live in a world that is largely shaped by human activities, which have altered ecological processes and ecosystems at massive planetary scales, with implications for the fate of biodiversity in the remnant ecosystems that we have today. Now, among the various factors that affect biodiversity, a lot of my work has focused primarily on habitat fragmentation so far, which is the breaking up of one's contiguous forest into smaller patches and the implications of such a breakage for the biodiversity in these in such landscapes. But increasingly, I'm also moving towards understanding the impact of climate change on how some of the biodiversity in human modified forests in particular might fare in a changing climate. So today, a lot of my work will focus on the habitat fragmentation work, but I will give you a brief overview of the climate change related work that I'm doing right now. I don't have too many results to share, unfortunately, so far, but where I'm planning to take my research in the direction of this interaction between fragmentation and climate change. So perhaps a lot of you might be aware that fragmentation is one among the top global threats to biodiversity. So you can lose biodiversity just because you break up once large pieces of intact ecosystems into smaller parcels where many species just cannot persist. But one major theme that has emerged or has been understood in fragmented systems is that the creation of forest edges often leads to changes in the patterns of diversity. For tree communities in particular, many long-standing field experiments and observational studies have shown that tree diversity tends to decline near forest edges. And these patterns have been studied from various forests around the world. But interestingly, the mechanisms underlying those patterns have not been investigated to such a deep degree. And often it was sort of qualitatively phrased in terms of the response to increased light availability at forest edges, that you have some species that do well at edges simply because there is a lot more light. Now, while that might be true in the very beginning, when a fragment is created or a new edge is created, often it is seen that even when there is a wall of vegetation that comes up, or if the canopy recovers at edges, you still don't have edges doing quite or behaving quite the same way or having quite the same diversity as interior forests. So that puzzle still remains about what are the mechanisms that seem to be driving changes to patterns of diversity, primarily a loss of diversity that has been extensively and commonly observed worldwide. So this is sort of a question that fascinated me at some point. And that got me thinking, one of the reasons why we might not be having such a great handle, or there might not be too much insights into the mechanisms driving diversity, is because a lot of the work in fragmented forests appears to look at the older life stages of trees. And from a whole body of work, parallel body of work in intact forests, we find that the mechanisms that maintain diversity, that shape non-random processes of survival and mortality in tree communities at least, appears to be strongest during the early life stages, that is seed-to-seedling transition, seedling establishment, and seedling survival appear to be particularly important and often leave long-term signatures for what the patterns of diversity look like even at later life stages. Now, among different processes that regulate these non-random patterns of mortality, one important or key process has emerged to be what or is known as what we call conspecific negative density dependence. Now, for those of you who are unfamiliar with this big mouthful of a phrase, all it essentially means is that when a species or as a species becomes more abundant in a community or locally, it experiences a greater limitation of itself. So what happens is that the population growth rate of a species declines as it becomes more abundant. And one of the ways that conspecific negative density dependence is mediated is by natural enemies. So think of it maybe in an epidemiological framework, which is that as you have more and more hosts, you're more likely to contract disease, and therefore you're more likely at a per capita level to die. Now, the role of natural enemies in mediating CNDD as conspecific density dependence is the acronym for it is known as the Jansen-Connell hypothesis. Now, back in the 1970s, both Jansen and Connell proposed that if you have host-specific natural enemies like insects or fungi, which means that each enemy on average has a larger impact on one host or one plant species than on other plant species, then what it does is that as the density or the abundance of that plant species increases, it brings down, or rather it increases the per capita mortality rate of that host species and brings down its population growth rate. And this kind of self-limitation that is imposed by enemies through CNDD allows or prevents any one species from occupying all of the available space for recruitment, for example, allows rare species to persist and therefore plays a key role in maintaining plant diversity in ecosystems that we see. Now, a logical corollary to this would be that if the impact of enemies is key, particularly host-specific impacts are key to maintaining diversity and helping rare species persist, then in any situation where this effect of enemies weakens in their ability to regulate plant populations, you should see a decline in diversity, right? So this was the basic premise that I followed or extended into forest edges, or what might be happening to diversity at forest edges. Now, some of the literature, which was completely unrelated to plants and that was just on fungi and insects, did show that edge effects occur for these groups of enemies as plant enemies as well. And often it is seen that specialized insects and fungi can be replaced by generalists. And these are not necessarily host specialists. For insects, there might be some host specialists, but also like habitat specialists. And this is something that I'm planning to follow up on at some point to look at what specialization really means. But overall, if specialized enemies are replaced by generalists, or if abiotic conditions such as warmer, drier conditions decrease the efficacy of some enemies or allow some particular plant species to escape the effect of enemies and become recruit in more abundant numbers, that might explain why diversity decreases or abundances shift towards particular species at forest edges. So to bring it all together, basically what I looked at was, do edge effects modify how insects and fungi regulate plant recruitment and diversity during these early life stages? And might that explain why we are seeing low diversity in edges? So I examined this question in a typical human modified biodiversity hotspot in the Western Ghats of South India. The Western Ghats is a 1,600-kilometer long mountain chain that occurs along the west coast of peninsular India. I conducted this study somewhere in the middle, which is a wet tropical forest. This particular landscape was a mix of tea plantations, some human settlements near the tea, and forest fragments arrayed across about 30 square kilometers or 35 square kilometers. And here I set up my sampling stations or sampling at different locations across the landscape. At each location, I set up replicated stations at the forest edge, going from the forest edge up to the forest interior, with stations at with replicates situated at 0 meters or between 0 to 5 meters, 20 to 30 meters, 50 to 60 meters from the edge, and in the interior forest in these conditions, which was at 90 to 100 meters. Each station, replicate station, consisted of 2 seed traps, which was meant to sample the seeds that come into this particular location in space, and 5 seedling plots. Now, why 5? Because we wanted an idea of the natural recruitment that is taking place, and 3 of the 5 plots were sprayed with an insecticide, a fungicide, and both pesticides together to see what happens to diversity, seedling diversity, for a given diversity of seeds that come in. What happens to seedling diversity when we are controlling the impact of these natural enemies? So we went out and set up these plots all across the landscape, covering about 35 square kilometers of the landscape. We monitored seed rain and seedling recruitment into these traps for about 2 recruitment seasons, a little over a year, which is approximately 2 recruitment peaks. And at the end of it all, we had this, in the beginning, we had this bunch, we had tagged a lot of seedlings, we knew what the diversity is like. And then in the end, we also had all of the new recruits tagged to know what the final diversity of seedling recruits looked like. And just for those who are unfamiliar, recruits just means the new seedlings that came up, which were not there at a particular location at that time. From the seed rain that happened, what were the new seedlings that came up are usually known as recruits in the parlance of our work. So I use this data to assess the question of do edge effects weaken the role of insects and fungi in promoting plant seedling diversity? So first of all, we looked at what were the patterns of diversity when we moved from the forest edge to the interior. And we looked at this for seeds and seedlings. In this graph, on the x-axis, we have the 4 categories at which I had set up my sampling stations, which is at 0 to 5, 20 to 30, 50 to 60, and 90 to 100 meters from the edge. On the y-axis is an index of diversity. Here I present results for inverse Simpson diversity, but we used multiple diversity indices to cross-check our results, and the patterns were fairly consistent. Panel A shows you the results of diversity for seeds, and panel B shows you for seedlings. Now, when we look at panel A, we find that seed diversity is not very significantly different as you go from the forest edge to the interior, even though there seems to be a declining trend in that the edges appear to have slightly higher seed diversity. But when we look at seedlings, we find that there is a stark difference in the diversity of seedlings up to about 60 meters from the edge compared to the diversity of seedlings we see in the interior patches, where the interiors appear to have significantly higher seedling diversity. So clearly something is happening where for a given diversity or a similar diversity of seeds, we have a decline, a sharp decline in the diversity of seedlings that eventually establish for a diversity of seeds that comes in. So the next step was to look at, is this diversity of seedlings in any way associated with the impact or the role of insects and fungi? So we compared the data that or the diversity of seedlings in our control plots with the diversity we found in the insecticide, fungicide, and the plots with both pesticides together. Again, in this graph, on each panel gives you the results for one category or distance category from the edge. On the x-axis are the 4 treatments: control, insecticide, fungicide, and both pesticide. Y-axis is inverse Simpson diversity of seedlings. And what we find is that up to about 60 meters from the forest edge, insecticide and fungicide did not seem to have an effect in shaping seedling diversity. But when we go into the interior forest, the diversity of seedlings that is observed in control plots declines significantly in plots sprayed with insecticide and fungicide. And this suggests to us that while these enemies are playing a key role in the interior forest, somehow their ability to regulate diversity appears to be weakening at forest edges, or up to about 60 meters from the forest edges. So the next question is, would this happen because there is a shift in their ability of insects and fungi to regulate the population dynamics during seed-to-seedling transition? Of course, I use population dynamics in a very, very broad sense, but the rates of seed-to-seedling transition, and in particular, how do insects and fungi shape the strength of density-dependent mortality during this phase? Now, I calculated density dependence using a functional form where the seedlings that recruit in a plot are modeled as an exponential function of the seeds that fall into that plot. I did this for all species and evaluated or got a community-wide strength of density dependence. And we will come to it at some point, how exactly I calculated it, but I can talk more to those who are interested in the details of that model. But in this graph, again, I show the results of density dependence in control plots versus the treatment plots for each distance category from the forest edge. On the x-axis are the different treatments, on the y-axis is the strength of density dependence. Now, what you need to note here is that the more negative or the smaller the value of the y-axis, the stronger is the density dependence. And theory suggests that stronger density dependence should be associated with higher diversity or maintenance of diversity. And corresponding with the results of diversity, what we find is that up to about 60 meters from the forest edge, the value of conspecific density dependence was getting stronger as we go into the interior forest. And at the forest edge in particular, it seems like the insects and fungi did not alter density dependence to a very substantial degree or to a significant degree as compared to other distances from the forest edge. And in particular, the effect of insects and fungi on density dependence seems to be strongest inside or in the forest interiors, but also there seems to be some ongoing effect in the middle areas as well. So we read this to, or we interpreted this to mean that the loss of diversity that we are seeing, at least at the forest edge, seems to be in some way correlated with a weaker ability of insects and fungi to regulate density dependent mortality at the edge. Now, why this is happening, or the exact mechanisms, pathways for why this might be happening, is not something that this study could tell, but it's something that I'm planning to go into in the near future, where we are going to look at plant soil feedbacks and how soil from the edge and interior affect conspecific and heterospecific soils from the edge and interior actually affect plant performance compared when you're looking at different species that recruit at different parts of this forest. And what I presented to you previously was like a community-wide mean of the density-dependent effects. But we know from a lot of previous studies that density dependence also varies from species to species, which means that some species are better able to tolerate larger conspecific densities or higher conspecific densities than other species. Now, if you're more tolerant of conspecific density, or there's a situation where your conspecifics, you are not as sensitive to conspecifics as you would be in another situation, would allow your abundances to increase. So we took a bunch of species that represented different life history strategies, particularly with respect to their ability to survive, with ability of the seedlings to survive in high versus low light. And we checked whether density dependent mortality during the seed to seedling transition stage varied for these species with respect to the ability of soil fungi in particular to regulate their populations. So in this experiment, we set up, we simulated densities or different densities, low and high densities of these species. We started with 6 or 7 species, only 4 of them actually germinated and yielded enough results for us to analyze, unfortunately. But these 4 species, which were set up at high and low densities in paired plots at the forest edge and the interior, gave us some interesting results. And the first thing that was apparent was that species are doing different things, which is what we kind of expected, right? And among these 4 species, you have Tuna ciliata, which is an extremely light-loving, typical pioneer species. Macaranga peltata, another typical, pioneer species, but with slightly larger animal dispersed seeds. Olea dioica, which does well in many fragments, but does well both at the forest edge and the interior. And Heritiera, which seems to, in general, do better at shaded understory conditions. So broadly, these 4 species spanned a range of seed sizes and life histories in terms of their ability to survive in low versus high light condition. And on the x-axis, the light and the shade basically mean light means plots at the forest edge, and shade means experimental plots in the forest interior. On the y-axis, we have the percentage of seedling recruitment or the germination success of each of these species. And what we find very broadly is that the results are mixed, of course, as most results seem to be. But for something, Tuna ciliata showed these typical patterns of density dependence with respect to soil fungi, in that when they were at the forest edge, their recruitment, whether at the forest edge or interior, their recruitment rates were always higher when they were at low densities. This high recruitment rate was, fell remarkably when they were in the forest interior, but was saved or was improved when we sprayed fungicides when they were at low densities. And a similar pattern was seen at high densities also, which suggests that a species such as tuna is not only does it require light for successful germination, but it seems to show these classic density dependence patterns, which appear to be mediated by soil fungi. And it would appear that the success of this species, or the lack of success of this species in the forest interior with respect to its germination, especially, seems to be driven by its susceptibility to soil fungi. On the other hand, we have Makaranga, another pioneer species, which really didn't show too many differences. It seemed to be sensitive to light, in that it needed highlight, nothing survived in the shade, nothing recruited in the shade, but it didn't show such strong responses to fungicide in particular. Now, we don't know whether that was because such few seedlings emerged that the fungicide effects just didn't come through, or if something else is going on there. But it does look like, rather than fungi, light is most important for this pioneer species. And these two larger seeded species, on the other hand, were showing mixed results, where for Olea, you find that in low density plots, fungicide did improve its survival, both in light and shade. But the effect of fungicide at high density plot only appeared to have a positive effect when it was in high light conditions. Now, we're not sure why this is quite happening. But these graphs sort of tell us that when we are looking at the effect of density dependence in regulating species recruitment rates, we do need to have experimental tests of what species of different life histories, and particularly with respect to their response to soil fungi or soil pathogens, are doing in these different combinations of environmental conditions that are found. Or in other words, that environment can mediate the strength of density dependence that you see in communities, and the ability of density dependent processes to regulate population dynamics or recruitment dynamics. But these results also made us think that, okay, should we be looking at trait-based recruitment at a much larger scale for the entire community that we have? So one of the questions, of course, was that, if you have these life history differences in the kind of species that are recruiting at forest edges, can we get a signature, or are some of these life history differences also related to the impact of insects and fungi on the recruitment success of these species? Because we know that some traits like seed size, for example, or a specific leaf area, which is basically the leaf area per unit gram of biomass, are related to the ability to be defended against enemies, or successful recruitment rates, investment in defense versus growth, etc. So we used 4 key traits, or 4 groups of traits: size, a bunch of leaf traits, and wood density for the older life stages, to see whether species with certain traits show differences in their recruitment patterns with respect to the role of fungi and insects in regulating their recruitment. And I present here the results for seed size, because those were the clearest, or rather, that was the trait for which we got clear results, and which is kind of expected, because seed size appears to be a trait that is closely correlated with recruitment or germination success in general, at the life stage relevant to the life stage that I examined in these studies. So in this graph, on each panel again shows you the results for each distance from the edge. Remember, it's 5 meters from the edge, about 20 to 30, 50 to 60, and about 100 meters in the interior forest. On the y-axis, you have the seed size, or the value of the trait for that species. Sorry, on the x-axis. And on the y-axis, you have the density of new seedlings that came up for that particular trait. And what we find is that up to about 60, and of course, the black line are the results for the control plots, and the blue and the red lines are the results for insecticide and fungicide treatment. And what we find that up to about 60 meters from the forest edge, we don't see much of a difference in the role of fungicide and insecticide in mediating differences in recruit numbers. But when we look at the interior plot, there seem fungi and insects do seem to play a role, a much stronger role in mediating which seedlings are more, or species of which traits produce seedlings in higher abundance. So it looks like in the control plots, by and large, or there seems to be a trend that larger seeded species are more abundant in the control plots as seedlings. But this advantage of larger seededness is lost if insects and fungi are controlled for. So this throws, out the question of, okay, is this role again of insects and fungi really what is driving the shift or also contributing to the shift at edges that we see in terms of functional composition of, or the dominance of smaller seeded species, sometimes a persistent dominance of smaller seeded species, even when the canopy gets covered up and light levels change. So again, something that I will be looking into in terms of its specific mechanisms in some of my future studies. And just like with with the taxonomic diversity, we looked at functional diversity as well to see whether changes in functional composition or the recruitment of some species is also associated with shifts in functional diversity. And yes, for seed size, we found that functional diversity of seeds is actually higher in the forest interior compared to up to about 60 meters from the forest edge. And again, the role of enemies appears to be the strongest in mediating functional diversity in the forest interior. However, one thing we found was that these functional diversity results did not really control for, or rather, when we controlled for species diversity using a null model, we found that the functional diversity results were not as strong as, statistically speaking, as what is seen in this graph here, which suggests to us that a lot of the functional diversity patterns are not directly driven by the traits themselves or are because of changes to the traits themselves, but might actually stem from the changes in species diversity that we had seen earlier. So to summarize, these results together showed us that it appears as though weaker regulation by natural enemies did correspond with lower diversity when it was in the forest interior. And this diversifying effect of enemies appears to be compromised as we go closer to the forest edge. And the decline in species diversity was also associated with a decline in trait diversity and changes in functional composition towards or might explain some of the changes in functional composition towards smaller seeded species that we see in forest edges. And one thing to look at in the future is, would this or might this loss of feedback between enemies and their plant hosts be a mechanism for persistent changes in the diversity of forest fragments, particularly with respect to edge effects in these human modified landscapes. And of course, one of the things that, always comes to the fore when you're doing these studies of recruitment is that, does it matter for diversity at remaining life stages? And this is something that I teamed up with, my colleague at Yale University, Simon Stump, then at Yale University, Simon Stump, who is a mathematical ecologist, and we decided to use modeling tools to explore this question of how well early life stage dynamics might inform patterns of diversity later on for these tree communities. And this exercise was contextualized in modern coexistence theory. Which was sort of formalized by Chesson, Peter Chesson, in his seminal paper in Trends in Ecology and Evolution in the year 2000, but has been developed and advanced considerably over the next or over the last couple of decades or so. But in effect, modern coexistence theory says that diversity, or the ability, stability of diversity in a community, is a tension between fitness differences between species and stabilizing mechanisms such as conspecific density dependence that allow species to self-limit. Self-limitation is crucial for rare species to persist and or prevent species from going extinct and locally extinct in communities. Now, fitness differences or larger the fitness difference, the more likely is it that species will go extinct unless there are strong stabilizing differences to counter those stabilizing mechanisms to counter those fitness differences. So this was the framework in which we looked at the recruitment process and the change in density dependence during recruitment process for diversity at later life stages. Any process that might lead to an increase in fitness difference or decrease stabilizing mechanisms is likely to reduce diversity in the long run. That's what we hypothesized. Now for this, we extracted fitness differences and strength of density dependence from the seed-to-seedling recruitment or the seed-to-seedling transition model, which, as I had mentioned to you, where J, the number of seedlings for a species, is modeled as a function, an exponential function of the number of seeds of that species that arrives at a site, as A times the number of seeds raised to B. Now, A is basically, it's the baseline seed-to-seedling recruitment rate, or in some sense, in each habitat, that would be the baseline fitness value or of that for that particular species. And here, I don't use fitness in the in the evolutionary sense, of course. I just mean recruitment success. Which is how it is in modern coexistence or used in modern coexistence theory. And the value of B is the strength of density dependence in during seed to seedling transition. Now remember that the larger the B, the weaker the density dependence. So when we put it all together for about 18, we had data for about 18 species to get these values at the species level. And here panel A shows the values of A for these different species as you go from the edge to the interior, which is a baseline survival. And you find that in general, baseline survival appears to be much higher for most species at the interior. And panel B shows you the strength of density dependence with smaller values or more negative values showing stronger density dependence. And again, like we saw with the community level patterns, interior forest species seem to, in general, experience stronger density dependence, but there's substantial variation at the species level effects when you're at the forest edges. We use the parameters, or we use these parameters to, these values to parameterize a mathematical model, a spatially explicit simulation to understand how seedling dynamics then bears upon the eventual diversity of trees or older trees that you see in a community. And how this model worked was that the parameters, the seed to seedling transition life stage was parameterized using our data. And in the remaining part of the life cycle, we just used lottery competition to figure out which individual might succeed. Once that seedling establishes, given all the stuff that's happening with seed to seedling transition, how well does that individual become an adult? And what does that mean for the eventual adult diversity? And in modern coexistence theory, one of the key parameters that tell you whether or not species are likely to coexist in a community is what we call the invader growth rate, which essentially means that what is the growth rate of the species, the ability of the species to invade a community. This is not invasive species, I said, but to invade a community of other species. Or in other words, what is the ability of the species to have a net positive growth rate when it is at extremely low density. And we modeled the invader growth rate using the parameters of that we got from the seed to seedling transition models. And we modeled this for 3 different scenarios where we had fragments which were subject to edge effects. We had just patches of similar size but which were only interior forest, or which we said were similar to intact forest. And then we had a fragment that was without interiors, where only, it was just edges, but we had different sizes. And those are the 3 scenarios that are represented in panel A, B, and C respectively. On the y-axis is the invader growth rates. If invader growth rates are above 1, it means that the species will stay in the community or it will coexist along with other species. Now, what we found was that fragment size always played a role in that in promoting coexistence. In that species that were smaller fragments, you always had species more likely to go extinct. This was definitely most prominent when you had fragments without any interior, where these stabilizing mechanisms were sort of playing out, or in other words, if edge effects compromise stabilizing mechanisms. And fragment size did not seem to have such a strong role in mediating or in mediating extinctions, local extinctions, if you're just talking about an interior or intact patch of forest where these stabilizing mechanisms were still operational. Um, so in effect, it means that it shows that there's something about habitat quality in terms of maintaining these mechanisms that promote diversity that are needed, in addition to just having these patches of forest make up a certain area um, um, of habitat for a community of interest. And in particular, it appears that if edge effects are compromising mechanisms of coexistence, stabilizing mechanisms, then it does compromise coexistence, long-term coexistence for the tree community in this landscape. We also matched up some of the data that we found, or the simulated diversity of tree communities, the composition of tree communities that we found with our parameterized models to the actual observed composition. And this was because we wanted to see, are we catching some of the dynamics? Like if we just use seed to seedling transition to model dynamics, do we catch, or can we represent some of the actual diversity or patterns of composition that we see in the forest today, that we observe in the forest today? And in this, in panel A is the similarity of the composition of simulated communities to the observed communities for adults, large trees. And in panel B is the match between simulation and observation for seedlings. And x-axis shows you the time through which these simulations were run, and y-axis shows you the value of similarity between observation and simulation, and how well it matched or how it changed through time. And it appears that, so if you just focus on the adults here, it appears that we are catching some of the transient dynamics of the forest, because clearly the similarity, using just that one life stage of seed to seedling transition, with everything else being a lottery competition, it looks like similarity between simulated and observed communities increases until about this dashed line, which is the time which indicates the average age of these fragments, what we think are the average age of these fragments. So clearly our simulations are managing to explain the composition that we see today in these fragments. And in some of the fragments, it appears that for at least some of these distances from the edge, we are going to see a change in fragment composition, or we're going to see a change in local community composition through time, which might indicate that we are sort of catching the transient dynamics, and that there are going to be future changes in the composition, even if just these changes in seed to seedling transition continue through time. So it looks like species coexistence or mechanisms of species coexistence is playing a key role, or might be playing a key role in shaping the future of diversity in fragments, habitat fragments. It does look like there is a relative shift in the importance of mechanisms between fitness differences and stabilization when we move towards forest edges, where it looks like weaker stabilization will affect diversity. So one thing to look at would be, what are the ways or that stabilization weakens in habitat edges. Using the framework of coexistence also tells us that we can probably assess habitat quality or compare the value of quantity versus quality in terms of these processes that maintain diversity, rather than just look at the outcomes in terms of species richness or diversity itself. Like, don't just look at the outcome, look at the processes that lead to these outcomes. Which I think should be key, or would be key, to formulate appropriate management plans for conserving biodiversity in many of these human modified landscapes. And just before I sign off, I mean, I spoke a lot about the biotic interactions that we were talking about, and I phrased or contextualized a lot of my arguments in saying that most people, or mostly, changes in fragments have been looked at in terms of abiotic effects of changes in light levels. So we did account for this, just so to let you know. And indeed, canopy openness, which is strongly correlated with light availability, is slightly higher near forest edges here. But there was no correlation between, or canopy openness did not play any role in explaining spatial variation in seedling diversity or functional diversity. And it looks like, at least in this forest, it is not canopy openness per se, but something else to do with enemies at edges that seems to be correlated with the patterns of seedling diversity during these early life stages that we are seeing. And the reason why I put this out to you is because that is not to say that abiotic conditions don't matter. Now, there is ample evidence, or increasing evidence, I would say, from many different parts of the world, which show that soil moisture or drought conditions are very important in shaping the distribution and composition of tree communities, particularly during the seedling life stage in tropical wet forests. In this study by Engelbrecht et al., they found that as drought sensitivity of seedlings increased, as indicated in the x-axis, their abundance in dry versus wet sites decreased considerably, which means that drought intolerant species declined in dry areas and drought tolerant species dominated in the wet areas, or increased when they went from dry to wet areas, which makes me think that the ability of seedlings to recruit and survive, or their fitness in fragments due to edge effects, might also be regulated by soil moisture, in addition to, or perhaps more than, just light itself. And indeed, some of my own work from the Western Ghats at very large scales across the Western Ghats shows that as we go from a gradient of low seasonal drought in the southwestern Ghats to high seasonal drought in the northwestern Ghats, there is a stark difference in species composition that is explained by the differential response of species in their sensitivity to these seasonal drought conditions. And we have also taken, we conducted this analysis for like 160 odd species, but we took about 15 to 16 of them, and which occur at both, either at the seasonal, at either ends of the seasonality and somewhere in between as well, to check how whether they show differences in their response to drought. And the preliminary results appear to suggest that mortality is not correlated so much with drought, but seedlings that are from drought or low seasonal drought areas, those species actually show a sharp decline in their photosynthetic rate and their growth rates when subject to drought for about 3 to 4 months, which means that they cannot handle seasonality, which suggests that if patterns of seasonality shift, which is happening in the Western Ghats, if dry seasons become longer or drier, then those are likely to affect some species more than others. And then the question that I'm interested in is that if we know that edges tend to be warmer and drier than forest interiors, then will the impact of these changes in seasonal drought, or just the drought that we see today itself, is it going to be stronger at edges? And therefore, these interaction between climate and land use will shape the kind of community that we see in the future. And can we scale, or I would be interested to scale from these local processes to landscape level effects of what kind of trees or tree species are likely to do well in a human modified landscape based on these changes, and which is also in some sense contextualized in the framework of modern coexistence theory, which is that are edges leading to changes or increasing the fitness differences, because, or especially when they're in a changing climate and subject to these multiple interactive stressors. So we have set up some of these initial experiments in the forest, which I hope very soon to visit and check, which was delayed due to COVID, but we are going great guns now. So hopefully in a couple of years, we should have some interesting results to share to understand what the effects of fragmentation or edge effects look like in terms of its interaction with an imposed drought for these different tree species. And with that, I'm ready to take any questions, this is, it's sort of, looking at biodiversity in the Anthropocene is a huge challenge. And every time I do one study, I feel like there's just so much more to understand. There's so little that I understand. But it's been fun. And I hope that in the near future, we can get stronger, better insights, use ecological theory to get stronger insights into what is happening in these human modified systems, to come up with actual theory-based, evidence-based management plans for human modified systems.